Hello, and welcome to this lecture on biological research ethics when we're taking a look at HeLa cells. Now, HeLa cells, as we're going to see hopefully over the next 15 or 20 minutes, were essential in the development of modern cell biology research. So to start out with, we're going to look historically. Ideally, when we're studying cell biology, when we're studying the kind of function of the human body, we want to try to figure out what's going on within the cells. And so the ability to study cells outside of the body is an incredibly important tool for biological research. Instead of having to, do on a complete experiment on an individual, whether that is a human being or an individual mouse or rat, the idea is that you can collect cells, grow those cells within the laboratory setting, and instead of working on an entire individual, entire human, entire rat, working on a plate of cells. And potentially it's possible to come up with hundreds and hundreds of tissue culture plates of cells from an individual donor, individual kind of dissection, so that you can end up with lots of subjects, lots of samples, lots of repeats, in a fairly efficient way. And so when we're taking a look at cell biology and we're looking at studying cells within a laboratory setting, what we're talking about is studies that are conducted in vitro. In vitro, Latin for in glass, in comparison with in vivo. In vivo is in life. And so if you're doing an experiment on an entire individual, you're doing it in vivo. If you're doing it on cells or tissues or organs in a culture situation, you're going to be doing it in vitro, in glass, or these days in tissue culture plastic that we can study. Now, when we take a look at these cells, again, looking back over 100 years, what we see is that the developments that we kind of take for granted within cell biology had to start someplace. So researchers at the beginning of the 1900s were attempting to study the cells of the body, but generally they would have to do it within primary cell cultures. With primary cell cultures, you basically start out with a tissue specimen that's been collected from an organism, whether that organism is a human being or a rat. You would take out a tissue sample, almost like a tissue biopsy. You would isolate it, you would grow it within a laboratory setting. But what they learned is that most primary cell cultures are going to have a limited lifespan. You're only going to be able to grow them in a tissue culture setting for a finite period of time. An example of that are embryonic chick fibroblasts. You can keep those going for about 4 months in culture, and it corresponds to about 30 cell doublings. So the cells are going to double basically every couple days, generate lots and lots of cells, but they're going to go through a process of senescence. They're going to start to age. Their characteristics may change as they get older and older. And ultimately, it becomes very, very difficult to maintain these primary cell cultures in a laboratory setting for long periods of time. And so the goal was to try to come up with cell lines. Cell lines would be collections of cells with, kind of a unique phenotype, a characteristic that we can study that are capable of growing indefinitely within the culture setting. So basically, you set up the cell line, and you basically have kind of an infinite amount of cells that are available to you without having to go back, do another dissection, or isolate the cells again to start all over again, like you would with a primary cell culture. And these were very difficult to find. About 1912, the first cell line was established by Alexis Carrel, who isolated embryonic chick heart fibroblasts. Fibroblasts are connective tissue cells, which are found within the embryonic heart. He was able to isolate them and grow them over a period of 34 years. And so, in essence, keep them going within the laboratory setting for an indefinite or infinite amount of time. But again, these were embryonic cells, and so there may be some kind of developmental characteristics based on that, which kind of contributed to the ability of them to live in that culture setting for a long period of time. It became very, very difficult to generate human cell lines. And so because of that, scientists in the early 1900s were really struggling to try to figure out, what is it about human cells that they won't grow for extended periods of time in culture, and what needs to occur for them to be able to grow in culture and be able to be studied. Now, taking a little bit of a side jaunt, we're going to look at an individual here. Henrietta Lacks was a tobacco farmer, and in about 1951, she was treated by Johns Hopkins Hospital for cervical cancer. So she went into the hospital. They determined that she had cancer of the cervix. They attempted to do surgery to remove it. They treated her with radiation. But again, this was a very primitive attempt because medicine at that point didn't understand cancer all that well and didn't have a cure for it. And she ultimately died relatively quickly within that same year, within 1951, died of that very aggressive cancer that had spread throughout her body. Now, as part of her treatment, a biopsy of the cancerous cells had been collected so that they could kind of identify what type of cancer she had, kind of study the cells at some point. But kind of excess cells that were kind of needed beyond for the diagnosis of her cancer, were used in a research project. And this occurred without her knowledge. And it's important to recognize that this happened, 1951, roughly 70 years ago. And the laws were very different at that time. And so the physician that did the biopsy was collecting the tissue and made it available for research studies. And this was legal at the time, although those rules have changed. We'll talk about some of those rules changing later on in this lecture. George Gay was a cell biologist at Johns Hopkins that was working on developing a human immortal cell line. And so basically isolating tissues from different biopsy samples, isolating the cells, and attempting to grow them for an extended period of time. And basically was fairly unsuccessful. He tried lots of different tissue culture media, chicken plasma, human umbilical cord blood, a whole variety of other natural and chemical kind of additives into the media to try to grow them for an extended period of time. He received a sample of Henrietta's cancerous cells and was able to determine that these cells, these human cells derived from cervical cancer, isolated from the biopsy, were able to continue to grow and divide outside of her body and served as a basis for what would ultimately become an immortal cell line. So if we take a look at this, he identified these cells as HeLa cells, H-E-L-A, derived from her name, the first two letters of Henrietta, the first two letters of Lacks, giving us HeLa. And what he was able to show is that these HeLa cells were able to divide relatively rapidly within culture, propagate rapidly, so undergo lots and lots of cell divisions, rapidly fill up the tissue culture plate that they were working with, and they showed no signs of cellular senescence. So previous attempts had shown that, human cells might grow well for, a couple of weeks or a couple of months, but over time, they're going to start to change, they're going to start to become old, and they're going to stop dividing. These HeLa cells didn't show that senescence, and they continued dividing, and they continued going, as if they were the original cells that had been isolated. Now, Gay had also been able to develop a way to ship these cells. Basically, he was able to take these cells, put them into a special tissue culture media, and then freeze them, and basically take these cells frozen, put them in a freezer, or ship them on ice to different locations. And once they got to the new lab at a new location, the cells could be thawed out and then used for biological research. And so this was really exciting. They could freeze cells and store them up for a period of time to use in the future, or they could distribute it to other scientists. And so we could have scientists around the country and around the world studying the same types of cells. So studying all of these HeLa cells, putting them through a whole variety of experimental manipulations and then comparing the results, again, because they're working with these same cells. If we take a look at what the HeLa cells have been able to do over the last 70 years, we can see that they contributed to many important scientific accomplishments. The HeLa cells were used in the development of the polio vaccine through the 60s, and more recently studied for understanding and treatment of leukemia. The study of AIDS in the 80s and 90s took a look at HeLa cells. They took HeLa cells into outer space to take a look at the effect of zero gravity on cells. So these cells have been used very, very extensively within the scientific community. In 2010, when these HeLa cells were looked at and Henrietta Lacks was looked at from a historical perspective, as of about 2010, there were over 60,000 scientific articles that were published on research with these HeLa cells. So again, huge impact on the understanding of cell biology. And it's estimated that in the 10 years since then, there are about 300 new papers each month that are published that are working with HeLa cells. So they were incredibly important and remain incredibly important for the study of cell biology. Now, with the advent of the ability to do genomic sequencing, DNA sequencing, there was a real interest in learning about the genes and chromosomal anomalies associated with these HeLa cells in order to try to understand how it was that they were able to be isolated and grown for such a long period of time. And so a group, actually two groups in about 2012, 2013, subjected these HeLa cells to full genetic sequencing. And they were able to come up with a full map of these cells, of the DNA of these cells, and were able to determine that there were kind of many things going on, a lot of variation in these cells, including things that impacted on the cell cycle. Again, if you've got a cell that continues to divide, you're thinking about the cell cycle being involved, as well as DNA repair mechanisms. And so the DNA repair mechanisms are thought to have an effect on preventing these cells from senescing or growing old or having limitations in their ability to divide over a period of time. They also did chromosomal mapping and found lots of deletions, tandem duplications, inversions, and translocations. And so kind of the genetic information in these cells is very, very different from kind of a normal, whatever a normal kind of human genome is going to be. And this likely contributed to the fact that this was a highly invasive cancer, as well as a cell line that's been able to grow in laboratories for almost 70 years now at this point. Now, the problem with doing this genetic sequencing of this individual, in this case, we've got Henrietta Lacks, and this with a little bit of a family tree going on with, her children, what would be her I'm sorry, not her great-grand, her children and her grandchildren now at this point, some relatives loosely related to them, cousins. So if we study the genetic information of Henrietta Lacks, we're also likely to be coming up with at least some of the information that is related to her relatives. Again, her descendants are going to have her DNA, which is passed down, through that process of meiosis and fertilization, so that her offspring would be carrying, a lot of the genes that were present within the DNA sequencing that was done. And what we know is that we need to be protecting private medical information. The HIPAA, the Health Insurance Portability and Accountability Act, protects an individual's health information and says that it can only be used for medical purposes, and it needs to be protected. More recently, in 2008, we had the Genetic Information Non-Discrimination Act that prevents discrimination based on the findings of genetic testing. But potentially with the publication of the genetic sequence of the HeLa cells, this could potentially identify information about the DNA and about the potential future health impact of her descendants. And so because of that, very early on, right after the genetic sequence was published, it was pulled back and made private. And about that time, NIH Director Francis Collins, you can see here in the middle. This is Francis Collins in the middle of this photograph with some of Henrietta Lacks's descendants, her children or grandchildren in this photo. They reached an agreement that would allow for the usage of the genetic information from this gene sequencing, but it wasn't wide open. It wasn't available to everybody, in the world. It was only open to those scientists that received approval from an NIH committee prior to the use of genetic information. So you couldn't just, use the genetic information in a voyeuristic fashion to try to kind of study it and kind of figure out what's going on with their descendants. You had to have a real scientific reason to be taking a look at that. And so you've got this NIH committee, and in respect and recognition of the family and the impact that this could potentially have on the family, 2 members of the Lacks family sit on this committee who decides who it is that has access to the genetic information from these sequencing. So if we take a look at this information, the HeLa cells have HPV-18 DNA inserted into them at multiple sites. So HPV-18 is a very aggressive form of human papillovirus that can go in and alter the DNA and alter the normal control mechanisms associated with the DNA and contribute to a cancerous setting. If we take a look at the HeLa cells, many of these cells would have an abnormal karyotype. If you take a look at the chromosomes, most of the cells are going to have between 76 and 80 chromosomes. And keep in mind, normal cells, normal human cells are going to have 23 pairs of chromosomes for a total of 46 individual chromosomes. And so the karyotypes that you see over here, we've got some, instead of having 2 copies of a chromosome, we have 3. In this case, potentially have 5 of them up here. Translocations, so that, this chromosome here is missing a portion of it, and it's been moved, to some other location, to some other chromosome. So they've saw lots of abnormalities with the chromosomes in the HeLa cells. And as they studied the genetics, they also found that these HeLa cells produce telomerase. A telomerase is basically an enzyme that goes and prevents the shortening of the telomeres. Telomeres are kind of a region on the end of the DNA molecule that gradually becomes eroded away with each successive mitotic event, each time that DNA is copied and replicated. And the idea is that with senescence, those telomeres are kind of eroded away to the point where once they're completely gone, those cells can no longer divide, and they become old and essentially die. Now, if we take a look at kind of going back to Henrietta Lacks and her family and the impact of these HeLa cells, what we can see is that Henrietta Lacks's family lived in poverty, in many cases maintained kind of an impoverished or challenging life, even though others were benefiting from the availability of her cells. And now her grandson, Ron Lacks, in 2020, and I don't have a copy of this book yet, I've just seen some notices of it coming out and starting to see reviews about it. Ron Lacks has written a book, again, her grandson has written a book telling her story from the family's perspective, and again, indicating that really they received no real benefit from these cells that kind of served as an advent for modern human cell biology, but took his grandmother's life. And as we look at the ones that have benefited from it, neither Gay or Hopkins profited from the cell lines. Both Gay and Hopkins University offered the cells freely and widely for scientific research, and kind of as we saw on that slide, contributed to kind of a major advances within sciences. But there were a number of companies, essentially just cell culture companies, that would grow the cells and sell them to researchers that didn't want to spend the time or the effort growing the cells on their own. And so some companies were making millions of dollars on the derivation or the use of these cells from her cervical cancer. Now, in about 2010, there was a book written by Rebecca Skloot, The Immortal Life of Henrietta Lacks. It got a lot of attention. It spent about 75 weeks on the New York Times bestseller list. More recently, in 2017, Oprah Winfrey did an HBO movie on Henrietta Lacks and her life and the impact of these HeLa cells. And so a lot of attention has been given to her family, and to the impact of these cells. And about that same time, they established the Henrietta Lacks Foundation, which has been helping individuals that have made contributions, very important contributions to scientific research, without personally benefiting from this. Particularly those that were involved in research without their knowledge or consent. And there have been funds that have been placed into this. They collect donations as well. And they make grants to kind of the descendants of Henrietta Lacks to provide for health coverage, or provide opportunities for them, scholarships for them to go to college and advance their lives in that way. Johns Hopkins as well is really reflected on their role in the creation of the HeLa cells, and are attempting to come up with ways in which they can honor the legacy of Henrietta Lacks. They've established an annual research symposium, the first of which looks at ethics in medical research, that is focused on an audience of scientists, clinicians, and ethicists that come together and talk about kind of ethical issues and ethical issues related to kind of the use of research subjects, and kind of advancing research subject rights kind of going forward. They also have an annual symposium focused on Baltimore high school students that will expose them to biological research and the ethics associated with this, with the idea of helping to kind of promote research and biomedical research and kind of biomedical careers to students in what would have been Henrietta Lacks's community. Hopkins is also providing scholarships, science scholarships for students in the East Baltimore region. And then most recently, they're looking at starting construction on a multidisciplinary research building on the Hopkins University campus that will ultimately be named after Henrietta Lacks. Now, if we take a look at kind of the rules regarding tissue samples as they were in the 1950s and current practices, we can see that they've changed pretty dramatically over the last 60 or 70 years. Kind of the major changes occurred in the 1970s. Taking a look at three basic areas of this. Informed consent is the idea that if you're doing a medical procedure, or you're doing tissue sampling or tissue collection, or you're subjecting a volunteer research subject to a research kind of paradigm or research procedure, there were no procedures for doing this in the 1950s. Now medical researchers have to provide detailed information to those patients and to those donors and to those subjects that are going to be potentially donating tissues or participating in research. In the 1950s, it was a very common practice to collect tissue samples from patients and then use those samples in a whole variety of ways. And now collecting samples for research purposes is under a whole variety of regulatory and legal standards, and has oversight of what's referred to as an institutional review board. And within this institutional review board, what happens is you have a group of individuals that are responsible for looking at research proposals and ensuring that they're being done in an ethical way that respects the rights and the autonomy of the research subjects. And then the final area we're looking at is using patient cells for research. In the 1950s, again, there were no guidelines, no local or national regulations for that. And now the practice is that the IRB, the Institutional Review Board, is going to review the studies using human participants or identified human remains or human cells and they have to make sure that the people that are donating these cells have been informed that these tissues are being collected and how the tissues are going to be used once they've been collected. Still, what we can see is that specimens that have been collected for specific study or for specific diagnostic test, in the case of Henrietta Lacks, they were collected to attempt to identify what type of cervical cancer she had. Many times they collect more than what is absolutely needed. And so these excess specimens, these extra specimens are often used for future studies, similar to the way that George Gay received access to Henrietta Lacks' cells when they were done with the diagnostic testing. Now, many of the informed consent procedures that individuals will receive when they're making a decision to participate in a study or donate tissues are going to describe how the samples can be used in the future. The default in many cases is that the excess samples or samples beyond what's needed for the immediate study or the immediate diagnostic test are usually available for future studies. Unless it's prohibited by the informed consent, in which case it would say something like the samples will be destroyed at the end of the study or at the end of the diagnosis or at the end of some period. And that's relatively uncommon. So there is, especially if you read the fine print, the opportunity to use these tissues in a whole variety of ways beyond what you're initially agreeing to in the first place. There is some limitations. The institutional review boards are going to say that if it's personally identifiable information, personal health information is included with these cells, it generally requires that the researchers go back and require additional consent. Having said that, though, that if they're anonymous samples, generally the IRBs are going to say that if it's cells that have been collected and they're no longer needed for their original use, you can use them if the information that comes along with them is not identifiable. And so, if you're using, Henrietta Lacks, a 31-year-old female cervical cancer patient generated these cells, it's one thing. If you refer to them as HeLa cells, which are named HeLa because it's an abbreviation for Henrietta Lacks, that then becomes identifiable and would require additional consent at this point. Now, the Code of Medical Ethics at the American Medical Association prohibits the use of human tissues for commercial purposes without the informed consent of the donor. And so kind of the extreme version of this is that if you take a look at human body parts, human body organs, we're prohibited for the sale or purchase of these organs. And so you can't go through and say, I need an organ, I will buy a match from somebody, because this kind of financial incentive could potentially be coercive to people, especially people with a lower economic base associated with them. And so there would be kind of a payment to them would have, in essence, an unfair kind of coercive effect on them. So we're prohibited from buying or selling organs, but many cell lines, genetic sequences, and biological products can be very financially valuable, especially if we look at the biotech and the pharmaceutical industries that, are generating billions and billions of dollars based on kind of the outcome of studies using these cells or using these biological kind of samples that may have been collected. It also doesn't talk about the value of the intellectual property that comes from manipulating the tissues or their products. And so there are oftentimes companies and researchers and universities will be issued patents for a specific use or specific application of cells and tissues that may have been given by a sample but have been altered or manipulated in some way to generate potentially a pharmacological drug or as a potential treatment for some type of disease or something like that. So with this kind of mini lecture, taking a look at HeLa cells, some questions to consider. Should patients have rights to their cells and their tissues after removal from their body? How does this balance out with the intellectual property rights of the researcher who modifies or develops the tissues into something of commercial value. Doesn't matter if the tissues to be used are somatic, in essence, if they're derived from adult body cells, or if they're germ cells, egg cells and sperm cells that have been banked for potential in vitro fertilization at some point in the future. How about if we're looking at in vitro fertilization embryos that are no longer needed? Should they be available for potentially scientific research, or should there be limitations? Now, ideally, if an individual is doing egg or sperm donation or going through the process of in vitro fertilization, they will have made their wishes explicit when they go through and they sign the paperwork for that. But again, this is a potentially gray area. Another question, should subjects be compensated in some way for the donation of cells or tissues? This would be beyond the minor honoraria that's often given in recognition of a donor's time and money. The IRB will often say, you can give your subjects or your people that are giving you tissue donations, cover their transportation events and give them, a Starbucks gift card or a Wawa gift card, kind of at a small level, kind of in recognition for their time, but not such a high amount that it would be seen as being coercive or potentially discriminatory. Again, if you're to the point of, I'm going to give you, $500 for a blood sample, people that, are, facing kind of uncertain economic times are going to see that as an incentive to participate in donation of the blood or potentially participate in a clinical research study in which they're going to, put their body at risk in order to receive some financial compensation. And so would large compensations be coercive or discriminatory against individuals from a lower economic status? So these are things to consider. A whole variety of references, and I'll post these on the Canvas site as well, if you're interested in reading some more background information about Henrietta Lacks, what's been learned about her and her family since that period of time, especially since 2010, when the book, The Immortal Life of Henrietta Lacks came out, have generated a lot of discussions and concerns about the use of human donated cells and tissues and their role within scientific research. Thank you and have a great day.